All right, so we're going to be going over cells and cell organelles today. So the reason why cells are important is because we know they're the smallest unit of life. And so I'm actually going to be writing, that's part of the cell theory. I'm going to be writing all of that down on this paper. So this paper, I was kind of toying on how to give you your cell organelle notes because it's all kind of a review and you don't need to know about all of the cell organelles. So what I did is I have this set of notes here and there's a bunch of tables on this set of notes. And so we're just going to go over the organelle. So I went through and listed every single organelle or every single cell structure that you need to know. So let's go ahead and get started, but first I want to remind you guys about the cell theory. Okay, so I'm basically going to be writing all over this page. So I don't know if you want to print out these notes and then have all the other stuff separate, or what it is you want to do. All right, so for our cell theory, we know that all living things are made of cells. Made of cells. Okay, so this excludes viruses. Viruses are not living, so they're technically not cells. They are cell-like, but they don't have the function of the cells. So all living things are made out of cells. The cell is the smallest unit of life. Okay, so every single cell has to be able to function. It has to be able to ingest food. It has to be able to absorb. It has to be able to excrete waste. It has to be able to form, cell functions. So lastly, our new cells come from old cells. Okay, so through the process of mitosis, all of our cells are going to be able to reproduce asexually. And so they're going to go through this process in order to essentially, like repair cells, replace cells, if you have an old cell that just needs to be replaced, or even a damaged cell, okay? New cells come from old cells. So that's a little bit of a review for the cell theory. You need to keep that in mind as we're going through this. So let's go ahead and talk about our structures and our organelles. So first off, we have cytosol. Okay, so cytosol is the semi-fluid substance in which organelles are found. So that is different from cytoplasm. Cytoplasm is the region between the nucleus and the plasma membrane. So it's also the interior of a prokaryotic cell. So the cytosol is the actual fluid substance. Okay, so it's the fluid substance, while cytoplasm is referring to the region. Okay, that's the major difference between the two. So we have just, what it actually is and then the area. So eukaryotic versus prokaryotic cells. Okay, so remember, prokaryotic cells and eukaryotic cells, for plasma membrane, we know that prokaryotic cells do have a membrane underneath their wall. Okay, so yes, under cell wall. Okay, eukaryotic cells, yes, they don't have a cell wall. Okay, ribosomes. Ribosomes are present in both. So I'm going to say yes and yes. The major difference, prokaryotic cells do not have membrane-bound organelles. So what does that mean? We're going to go over that here in a second. Eukaryotic cells do. The nucleus is membrane-bound. Okay, so because it is membrane-bound, prokaryotic cells do not have that, but a eukaryotic cell does. Also, eukaryotic cells are generally much bigger than prokaryotic cells. So something that we need to talk about, and whenever we perform one of our labs that's coming up, we're going to talk about surface area to volume ratio. As the volume increases, your surface area to volume ratio is going to decrease. So what does that mean? So if I look right here, I have these three cubes. One of them is 1 millimeter, the next one's 2 millimeter, the next one's 4 millimeter. So the volume is 1 millimeter cube. My surface area is 6 millimeter squared. Surface area to volume ratio is a 6 to 1. Okay, that's a really high ratio. That's what I want. When I compare that to over here, I have a bigger cube. Surface area is 96 millimeter squared. volume is 64 millimeter cubed, when I try to calculate that ratio, it's a 1.5 to 1. That means that all of this surface area, okay, so technically the surface area to what's on the inside of the cell is less. Okay, so down here where I have this small little cell, I have a very large surface area to volume ratio. So what that means is that anything that needs to be transported in or out of the cell will be able to do so more efficiently with a smaller cell. Okay, so smaller cells are more efficient to transfer materials in and out. Okay. And then also, smaller cells won't build up waste. Okay, it'll be able to transfer out. Okay. All right, so because of the metabolic requirements that cells have, you have a limit. They have to be able to absorb enough materials to sustain the cell and get rid of any waste products. So eukaryotic cells also have organelles, so that way those eukaryotic cells can specialize. Okay, so all of our eukaryotic cells have general organelles. So all these organelles we're going to go over, pretty much all of our eukaryotic cells have these organelles. There's a few exceptions, but some may have, some structures more than others. So let's go ahead and talk about some of these structures. Okay, so first off, when we're talking about genetic control, we're talking about our nucleus and our ribosomes. So genetic control is referring to housing DNA or expressing proteins. Okay, so we know our nucleus, okay, holds our DNA. Our mRNA is also made there. Has a nuclear envelope, that's our membrane, nuclear pores to release our mRNA, and this has all of our genetic information. So DNA is normally found as chromatin, and then it'll turn into chromosomes for division. So if you don't remember, chromatin is just like a big ball of DNA. Okay, so it's a big ball of DNA. Okay, chromosomes are my little fluffy X's. Ribosomes, those have 2 RNA subunits. Okay, I thought I'd get this done before the bell. Okay, so it kind of looks like a little muffin, a little upside down muffin, but that's what our ribosome looks like. And then its function is protein synthesis. Okay, so we have 2 different types of ribosomes. You have some that are free. These are proteins that are in the cytosol. So in our prokaryotes, they have free-floating ribosomes. And then you have some that are bound to the ER. All right, so next we're going to go over the endomembrane system. Okay, so endomembrane system, the reason why we say this, endo means inside, okay, and we know what a membrane is. So all of these have internal membranes, okay, so are folded membranes. Remember, one of the key things when we're talking about cells, they need to be able to have a lot of area in order for reactions to occur. And so what this does is it increases surface area. And so because there's an increase in surface area, that's so important. It gives a lot of space so that way reactions can occur. All right, so we're going to be talking about this. That's one of the key things for cells, when we're talking about the cell membrane and we're talking about cell functions and cell, structure and transport and all of that stuff. The fact that they have folded membranes means they can increase their surface area. So a good example, if we're looking at our ER, so we have a smooth ER and a rough ER. So really the difference is either the lack or presence of ribosomes. So our rough ER, we have ribosomes that are attached. Our smooth ER does not. And so, because the ribosomes are attached on the rough ER, The primary function is for protein synthesis. The smooth ER is going to be for lipid synthesis, and because of that, we're going to find that mostly in the liver. Okay, most of our ER is found in the liver. But if you remember what an ER looks like, you have, doesn't matter if it's smooth or rough, so here's your nucleus. You have all these little wavy things right outside the nucleus. Okay, that's my ER. All of that is the ER. And then if we're looking at it, really closely, it's really wavy. And so that gives us a lot of area, a lot of surface area for all those reactions to occur. If you need proteins produced, it can do that in mass. If you need lipids produced, it can do that really quickly. Same thing with our Golgi apparatus. Golgi apparatus is really, it's like a lumpy, blobby thing, okay? But because it's so blobby and it has all these little pieces to it, okay, so your Golgi apparatus has all these little flattened stacks of membranes. And so it's able to modify, sort, and essentially process anything that needs to come in or out. And so, when it does that, it's going to package them nicely. You have two sides for your Golgi apparatus. Your cis side receives any vesicles. Your trans side is going to ship vesicles. We don't quite need to know that, but I'll explain that when we do tomorrow's activity. Your lysosome. Again, it's a membranous sac, and it's full of enzymes. Remember, lyse means to break down. Oops, wow, I was writing the wrong type of break. Break down. Okay, I was writing about car brakes, apparently. All right, so this is for digestion. So anything that the cell is going to need, if it needs to perform phagocytosis, autophagy, apoptosis, things like that, the lysosome is going to be responsible for that. Lysosomes are only found in the animal cells. Plant cells do have a form of lysosome, but when we're talking in general, lysosomes are only in animal cells. Lastly, your central vacuole. It's a membrane-bound sac, and that's where all of your stuff is going to be stored. So animal cells do have vacuoles, like an adipose tissue where our fat is stored, but a central vacuole, okay, central vacuole is a big one, okay, that's only in plant cells. So again, we're going to continue with the fact that we have all these membrane folds for an increase of surface area. So if I look right here for my mitochondria and for my chloroplasts, they have all of these membrane folds. Okay, same thing with my chloroplasts. We have the membrane folds called thylakoids and grana. So both of these have a lot of surface area because they need to perform very special functions. Okay, so cellular respiration gives ATP. Okay, and photosynthesis gives glucose. Okay, we need those forms of energy. And so something else that's really interesting when we're talking about the mitochondria and the chloroplasts, it's a major evidence for endosymbiotic theory. And so essentially what that's talking about is how we had two advanced prokaryotic cells way back in the day with primordial soup, and one engulfed another. And one of the evidences for that is that it contains its own DNA. Both of them do. They have so many membranes. Remember that we're trying to increase surface area. That way we can get a lot of ATP and get a lot of glucose from that. A couple of other things. Cytoskeleton. Okay, cytoskeleton is used for mechanical support, help to maintain shape. And then it's also going to anchor anything that the cell might use for motility. So flagella, cilia, things like that. And it's also going to help organelles and enzymes to stay in the correct place instead of moving everywhere. Lastly, okay, so we have our cell walls. Okay, cell walls are extremely important. So when we're talking about cell walls, depending on what cell we're looking at, we're going to have a couple of different types. So cellulose, okay, that's my plant cell wall. Chitin is my fungi cell wall, and peptidoglycan is my bacterial cell wall. So y'all are familiar with cellulose and chitin. Peptidoglycan, okay, so peptide, remember I told you that was a protein, and then glycan is a carbohydrate. Okay, so we know that this particular structure is a carbohydrate combined with a protein. Okay, so the main functions for our cell wall are to protect, maintain shape, prevent water loss. And so, we know that there are several different organisms that have cell walls, plants, fungi, some prokaryotes, protists, things like that. Our extracellular matrix. Okay, so this isn't necessarily something that you need to memorize, but it's a word that gets tossed around in AP Bio just because we talk about it a lot. Our extracellular matrix are going to be things like glycoproteins. They're things that are external to the plasma membrane that are used for cell communication. And so cell communication is something that we talk about in AP Bio that we actually never talk about in freshman bio. So, this is just a heads up for something that's coming. So hopefully this helps. This was just a quick review on organelles. We're going to continue on with this. And remember, the important thing, we have a couple of important things. One, folded membranes have that increase in surface area. And then two, smaller cells are more efficient to transfer materials in and out. Okay, those are the two really, really, really important things from all of this. Let me know if you have any questions.